Hi, this is Amanda Savant, and today I will be covering the cell cycle. Specifically, we'll be talking about mitotic cell division. So to begin, we're going to start about covering a few basics about mitosis. And so mitosis is a type of cell division. It produces identical cells and it is essential for growth and development. And an example that I wanted to share as well that is important for mitosis is tissue repair. And so I'm pretty sure almost everyone has gotten a cut once in their lifetime before. And so usually, sometimes it's like by accident as well. It's like, oh, I got a cut, like I didn't realize that. But then, a few weeks later you might look back and it's like, oh, it's completely gone. It's not even there anymore. Well, that healing process is the fact that tissue is repairing, and it has to do with mitosis. And so I just think that's really cool. Also, we don't want to confuse mitosis with meiosis. Meiosis is also a type of cell division, but it produces something completely different. And so we're going to focus on mitosis and the production of identical cells. And so we're just going to move on into kind of like the cell cycle and looking at this quick overview. And so the cell cycle consists of three main parts, which is interphase, mitosis, and cytokinesis. Interphase has 3 parts to it, and mitosis has 4. And we will be going into each of these in depth as we continue on in this presentation. So to begin, we're going to start off with interphase. And so to begin, the very first thing in interphase is our G1 phase. So in our G1 phase, our cells are growing and functioning normally. Protein synthesis is occurring, and that just essentially means that proteins are being produced and being created, and so all of that is happening here in this first phase. And we can see a picture just of what the cell looks like. And I wanted to point out this chromatin thread. And so our chromatin thread here is made up of DNA and protein. The specific protein here is histone, and that assists in just compacting the DNA into the nucleus here. And so the nucleus is pretty small, and if the histone wasn't there to help the DNA be small and to contract into the nucleus, then we would have an issue because our DNA, if we were to stretch it out, it is extremely long and it wouldn't fit in the nucleus if we didn't have that protein. And so I just wanted to point that out because that's important to know, and if you're looking at this, you might be like, oh, I don't know what that word means. And so we will also be talking about centrosomes later on, but definitely just notice how here there's 1 and then later on there's going to be 2. And so in our G1 phase, if there is a cell that is going to divide, then it is going to move into our synthesis phase. However, if the cell is not going to divide, then it's going to stay here in our G1 phase and it's just going to continue functioning as normal. And as we move into this synthesis phase, also known as the S phase for short, DNA replication is going to be occurring. And so we can see here from this picture, we have these first homologous chromosomes, and so that's what they are in relation to each other. And so these chromosomes are going to be replicated. And so we see after DNA replication, we have 2 blue and 2 of these, I would say it's like a magenta color. And so these two blue are going to be called sister chromatids, as well as these two magenta are sister chromatids. And our chromatids are going to be connected and joined together by what's here in the middle called the centromere. So that is what's holding these two together. And so DNA replication is a really important process in interphase and overall in the cell cycle, because DNA has to be replicated before the cell can go through the process of mitosis. And since that is so important, I just wanted to walk us through DNA replication real quick and talk about it and give a little background about it. And so DNA replication is semi-conservative. And so if we were to break that word up, semi means half, and to conserve something is essentially, to save it. So it's kind of like we're half saving something. And so that's a great way to remember that word and what semi-conservative means in relation to DNA replication. So when we look at semi-conservative, what we mean here is that there is going to be the old strand, that's the parent, that is the original strand, and then we are going to get this new strand over here. So semi-conservative is just that. There is an old strand as well as a new strand. So when you break down the word, that just kind of makes sense because it's half. We're half saving. So we're saving one part and creating one new part of the DNA. And so enzymes are essential for replication. And so as you can see here, there's these endings that are highlighted. It's like, why is that highlighted? Maybe that's just a mistake. But no, the reason for this is that I wanted to point out that enzymes usually end with ASE, and that's just a great tip to help you remember if there's a word that you're reading, it ends in ASE in a science textbook, it more than likely will be an enzyme. And so to begin, we're just going to look at helicase, and helicase we can see right here in our picture, and that is just essentially what unwinds our DNA. DNA polymerase is going to be considered the builder. So it replicates the DNA molecules to build a new strand. And so here we have DNA polymerase right here and then up top here. And so primase is going to be important because it is what makes the primer for DNA polymerase to know where to start. Ligase is going to be what joins our fragments here. And so it's really important because it's going to connect these shorter strands to create the full strand of DNA, which is completely necessary. And so when we go back up here, so the synthesis phase and DNA replication, we just walked through that, and it's really important because once our cell replicates, it has to divide. So it must enter this G2 phase. And this G2 phase is going to produce the proteins that are necessary for the cell to undergo mitosis. And the cell is going to check for complete DNA replication because we want to make sure that the that the DNA is being completely replicated, because we don't want to have any mutations or any issues as we go through mitosis. And as you can see here, two centrosomes have formed. It's essential for mitosis to begin. And from the beginning picture here, there is a lot more of this chromatin thread because our DNA has been replicated. And so our DNA is going to our DNA is going to begin to wind tightly around the proteins, which signals the start of mitosis. And so we're going to move into the phases of mitosis, which can be remembered by the acronym PMAP. And so to begin, we're going to look at prophase, and in prophase, DNA is going to begin condensing. And as the DNA condenses, the chromosomes are going to become visible. And our chromosomes are just the chromatin thread, but in a condensed form. And so we can see here, these are our centrosomes, and our centrosomes are going to develop these spindles. And as we begin, there's still going to be a nucleus and a nuclear membrane, but that has to go away, or our spindles are not going to be able to attach to our sister chromatids. And so in the process of prophase, our nucleus, nuclear membrane, and envelope, that is going to disappear and it's going to go away. Our spindles are going to begin to form, and as you can see, our centrosomes move to opposite sides of the cell. And so now we are ready to move into metaphase. And in metaphase, our chromosomes are going to line up along the middle of the cell, and this is can be called the equator, or I like to call it the metaphase plate. And so as you can see here, our spindles attach to our sister chromatid, and that's what happens in prophase. So they connect because they are going to help with the transition into metaphase by lining up the chromosomes along this metaphase plate. And so the purpose of this is just to make sure that when the cell begins to divide, that there is a copy of each chromosome in each of the two new daughter cells that are about to form, because remember, the cell needs to be identical. And if the cell doesn't have all of the chromosomes, then it is not going to be identical, and that will not lead to mitosis being complete. There would be an error. And so we will move into anaphase. And in anaphase, the two chromatids split. So you can see here we have metaphase. The spindles are attached to the sister chromatids. And so the spindles are going to begin to pull these chromatids apart. And also you can notice that in metaphase, it's very circular, but as the process of anaphase is happening, this cell looks a little more wider. It kind of looks like I thought of a watermelon. And so the reason that this is happening is as the spindles are pulling the chromatids to the opposite side, the spindles are also stretching the cell to make it bigger and get ready for telophase and eventually cytokinesis. And so in telophase, you can see that the separation is a lot more distinct. Our copies of our DNA are on complete opposite sides of the cell, and you can also see here that our nucleus is beginning to form around the DNA. Our spindles, mitotic spindles, have contracted, so they have gone away because they are no longer needed. And you can see here that the cytoplasm is slowly beginning to separate. Also, in telophase, our DNA is beginning to, our chromosomes are beginning to unwind, and so the DNA is going to be able to be less condensed. And then we move into our final stage, which is cytokinesis. And in this process, cytoplasm divides, creating our two new daughter cells. And so in an animal cell and a plant cell, these processes look a little bit different. In an animal cell, we have this cleavage furrow, which is just a contractile rings that separates and breaks the cytoplasm, but in a plant cell, it's the cell plate. But essentially, in both, the cytoplasm is being divided, and that is where we get our two new identical cells. And so here's just a little overview of the steps, which is just a really nice picture for us to look at. And then that's my works cited. I hope this was informative and helpful. And that is mitotic cell division.